Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. We invite you into the stories, struggles, and breakthroughs shaping the future of human health. From cutting-edge brain science to discoveries transforming how we heal, adapt, and thrive, we explore the ideas that matter and the people behind them. Together, we look across diseases, technology, and research fields to understand not just what is changing, but why it matters for patients, families, and the future we all share. Join us as we uncover new insights, spark collaboration, and illuminate the science that can improve our lives. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of health science. Joining me today is Dr. Daniel Gershwind. Dan is the Gordon and Virginia McDonald Distinguished Professor of Human Genetics, Neurology, and Psychiatry at UCLA. He also leads the Gershwind Lab and serves as Associate Vice Chancellor for Precision Health. He is a pioneer in neurogenics and transcriptomics, and his work has reshaped how we understand autism and other complex brain disorders. So welcome, Dan.
Thank you very much. It's my pleasure to be here.
Thank you so much for taking the time. You are very distinguished, very experienced, have done some amazing, innovative work, and I can't wait to kind of dive down and hear your thoughts. But let's just kind of start with, you have spent your whole career studying genes and how they shape the brain. What is the biggest misconception that people still have about genes and genetics and brain disorders?
What a great question. Well, I can't say what everybody's misconception is, but one thing that sometimes gets misconstrued is that because I study genetics, that I am a genetic exceptionalist, that, those of us who use genetics believe that everything is genetic. And obviously that's not true. All human disease has genetic and environmental components. It's just that genetics provides a tractable, studyable space that can identify causes. And then once you identify causes, allows you to lead to mechanisms in a way that is unbelievably efficient and powerful.
And so you started this very young in your career. You've helped pioneer some of the systems that, the biology systems and approaches to looking at these networks instead of single genes. And, what is that about looking at a gene network that is very different? What does it tell us that's different about the brain and the diseases of the brain?
Well, it's like any process, so think about it this way, maybe this is helpful. As we began to enter this period that's truly a genetic renaissance, where we're identifying hundreds, if not thousands of genes that cause a wide variety of biomedical disorders, from cancer to disorders like autism that can impact mental health and neurodevelopment, there's a challenge that comes, and we know that genes don't act alone. No organ in our body acts alone. No cell in our body really, truly acts alone. They're part of integrated systems. And so rather than looking at one gene at a time, we kind of reason that the genes that kind of play together are working together. And if they go together, they're likely to represent a similar biological function. And indeed, that's what we found. That we could identify networks of genes that were fairly stable, that represented all the different crucial biological elements of the brain, from organelles within cells, which are like the machines within the factory, to cells, to tissue, to protein networks. It's really kind of amazing, and that we had a lot more power looking at the way the things interact and change together than just by looking at one thing at a time, because these systems adapt. And so you might have a particular mutation, but it has consequences And there's also ability of the system to adapt and to be resilient. So looking at gene networks was really a transformative thing for us and for the field because it allowed us to make sense of hundreds or thousands of genes rather than looking at one at a time. And of course, looking at hundreds or dozens or thousands is a much more coherent way to look at things than looking at one at a time. And if you see hundreds of genes moving in one direction that represent a particular biological function, it means a lot more than just seeing one or two.
So I'm going to go back to what you first said about being, studying the genetics and studying genes and not, I don't think you use the word a purist, but, that you just don't focus on that. You're kind of looking at the outcome of the system of systems. I've had other discussions with researchers where they will say, well, this disease or this condition, there is a genetic component to it, but it may be only 18, 20%. The rest of it is what is happening inside the genetic process or the biology of the body. Is that what you're talking about? Is that when we study the network, we're actually getting to see a much more precision of what's happening versus just saying, well, that's your genes and that's your heredity?
Well, it's a little bit different than that. And so I'll try to address that. It's a very complicated issue. So the genetics is the one piece. It's the kind of causal anchor in a way. You have your underlying kind of genetic susceptibility to disorders. And that can be measured. And in some conditions, we have a much more precise view of the genetics, how much is heritable, how much is genetic but non-heritable. For example, Down syndrome is genetic, but it's not heritable. So you have different components like that, things that are inherited from the parents, things where there's a genetic mutation that hasn't been inherited. And we can estimate those in the population for many, many disorders. And for most mental health disorders, the heritable contribution is over 50%. And for things like autism, it's 80%. Same with schizophrenia. And those are pretty decent estimates. And so there may be other things like major depression, where the heritability is closer or maybe a little under 50%. And it's necessary to have environmental stressors and other things that cause it. So it's not unusual at all. So heritability and genetic contributions vary across the disease spectrum and especially in mental health conditions, which we don't really call diseases per se, that many of the serious ones that we're thinking about have striking genetic contributions. And so that made it very clear that there was likely to be gold there if we started to dig. And therefore, about now, let's say, almost 25 years ago, we started to dig in earnest. And that's been amazingly successful in autism. There have been hundreds of mutations identified in different genes, more than 200 genes that contribute to autism. Most of those mutations are not inherited. They're de novo mutations like the Down syndrome mutation. And those are more likely to cause these kind of early childhood onset disorders. In other neuropsychiatric conditions, such de novo mutations are rarer, especially those that have later onset, like schizophrenia. They still occur, but they're not as common as they are in autism, and they don't account for as large a percentage. They might account for 15 to 20% of autism, rare, kind of non-inherited or rare inherited Mendelian mutations. So then you go to something like major depression or bipolar, and there are very few of these kind of rare mutations. It's mostly common additive polygenic risk. And so that, yeah, does that make sense?
Yeah, it does. It does. So maybe if we can just step back and talk about where the state of our understanding of autism is, because I think there's a lot of misconception. You hear things like, well, on the spectrum, or you hear people joke about, being on the spectrum or not even understanding what that spectrum is. What is the state of understanding of autism and that spectrum of what that means?
Well, it is an incredibly broad spectrum when you look at things like intellectual functioning, for example, or language, right? There might be 25% of children might be nonverbal, but only about 30 or 40%, maybe a third, have intellectual disability. Most people with autism don't. So they're a very wide spectrum. And the diagnostic spectrum has broadened considerably over the last 3 decades. When I started working in autism, it was thought to be 1 in 2,000 to 1 in 5,000 children. But that's because it was incredibly narrow definition. And as the definition has broadened and clinicians have gotten better at identifying autism, it's clear that it's more in the realm of 1 in 100. So, over 20 years, autism hasn't increased, 100-fold. It's that our ability to diagnose it in what we call autism or autism spectrum. And, it can be helpful to be, to kind of understand, that spectrum. On the other hand, it's created some controversy because there are people who are on the very, let's call, high functioning end of the spectrum who don't need really any therapy. They may not even say they have a disability. Some of them may say, well, I have a mild disability that needs some accommodation. And others may not at that end. And then you have the very severe end, let's say the 30% with intellectual disability. And then even people without intellectual disability who have very severe autism, who are intellectually very high functioning, but have extreme difficulty in interacting normally. And, because of the autism, those people have a very different condition that needs to be medically dealt with and diagnosed and treated. And we have to kind of recognize that. And that kind of spectrum has created some of that controversy.
Yeah. Yeah. In the widening of the definition, has that allowed for more kind of precision of certain areas? Do you think that over time there'll be a narrowing so that there can be a deeper dive into some of
Sure.
these other areas?
Just like any other biomedical condition, eventually we'll use, because this has such a genetic liability, my sense is that genetics and clinical factors, both, will be kind of brought into models where we identify subtypes. The other thing is, if you have one of these rare forms of autism, which maybe almost a fifth of children do, or adults, that would be treated maybe very, very specifically, just like a cancer with a specific mutation. It's not cancer, but just the analogy is, now in people with cancer, we sequence their tumors, and in about 10%, we identify a mutation that that says, they have this, let's give this targeted therapy for that. EGFR mutations in lung cancer and certain, mutations in breast cancer, et cetera. Very, very specific treatments for that. And so the same thing in autism. When you know what the genetic cause is, you have a place that you can potentially target. And I'm very optimistic about that in many cases.
So the diagnostics, you said the diagnostics have gotten better. How have they gotten better? What is primarily used for the diagnostics? And is our public health system able to, in a broader way, be able to do this diagnostic?
There's just been better dissemination of tools, more education. I would say that's a major part of it. And then on the genetic side, the technology has moved. Genetics really isn't involved in diagnosing autism per se. It's involved in identifying kind of what form somebody might have to kind of explain the autism. And we're at the very early stages of moving from the genetics to treatments. but we are. And that's a process. It takes time. Even in yes, a blood test. Yeah, if you sequence the DNA, you do whole genome sequencing or whole exome sequencing. You sequence the genome and you can find in about 15% of people or so, plus or minus, a mutation that's likely causing, if not making, a major contribution to their autism.
So in the study in autism and getting that better understanding of some of the, the spectrum and some of the subtypes, what else are we learning? Because I think there's others that are looking at the connection to things like epilepsy or to other kind of conditions. What is your thought there, and what is your understanding as we get, better at that diagnostic, how we understand the broader brain?
Most of these mutations that are occurring in autism aren't specific to autism. They're basically genes that are very important in brain development. Think about it this way. The brain is developing along a track. It's pretty resilient, can have a little bouncy here and there. But if you get a big enough bounce, it bounces off the track. Or there's a famous developmental biologist named Waddington who coined the term canalization, like a canal. And there's like an energy slope where development occurs along the low energy, and you can be bounced around a little bit, but you come back. But if there's a perturbation that's severe enough, like one of these developmental mutations, it knocks you off the track. And most of these mutations that predispose to autism, many of them predispose also to intellectual disability or intellectual disability plus epilepsy. So they're not kind of autism genes in one way. They're genes involved in brain development that, when not expressed properly, predisposed to autism. And some of them predispose to autism much more than other things, and others predispose more to to intellectual disability, and autism is just a small part of that. So we're at a point where we are beginning to understand that, but it's a very exciting time, but we don't quite have our hands around it yet. I can say that a lot of the mutations that cause autism and epilepsy, but not all, a lot of them involve early brain development or involve things that control electrical signaling in the brain. So they make a lot of sense, right? If you affect the way a neuron fires, you're more likely to get epilepsy, which is a disorder of neuronal firing, etc. So, there definitely is a lot of understanding, and we're just at a point, like within the last year or so and moving forward, where we're beginning to understand subtypes better, how the different genetic forms contribute to them. And I really think over the next 5 or 10 years, that's going to be, a major thing, this precision medicine. For example, if we can diagnose somebody early, maybe even in utero, this is just for the future. Most of these mutations knock out one copy of the gene. So there's another copy. You have 2 copies of every gene, one from mom, one from dad. There's another totally normally functioning copy, supposedly in most cases. And so is there a way to turn up or to augment the function of the normal non-mutated copy? And the answer is yes. We've done some work, and some collaborators have, showing that we can use CRISPR activation without editing the genome to just turn the gene back on at a very early stage, and in vitro models, in a dish, rescue what is seen, including in some of these epilepsy syndromes where we can make neuronal firing normal again and kind of normalize things. Now, the challenge there is now we have to, to move that into a therapy, there's a lot that would have to be done. We have to figure out how to deliver that to the brain. We might have to figure out how to deliver that to the prenatal brain, right, in utero. That means we have to diagnose people then, or some of these may work fine postnatally as well. We just don't know that yet, right? So there's a lot of work to be done that's essentially, you could call it engineering. We need to figure out how to engineer these things to deliver them. But I think theoretically and even practically, there's a kind of roadmap for moving forward with this.
Yeah. You mentioned the neural firing. I don't know, and I don't want to put you on the spot, but a couple of weeks ago there was a paper in Nature that was published about, seeing for like superagers is neurogenesis, this kind of repair or regrowth of neurons. I don't know if you saw the paper, but
Yeah, I did. Yeah.
yeah. So what are your thoughts there? Because that sounds like some of the assumptions that we believed about the death of neurons in the brain, maybe kind of we're at that time where maybe they're not the right assumptions anymore.
I think the jury's still out on that one. There are technical issues and other things to think about.
Yeah.
So that finding's a little controversial, but I would say that in terms of what we understand about neurogenesis, it's also plausible. So it is plausible. So I think we have to keep an open mind.
Yeah. Yeah. So you were a part, I don't know if you were, you were the primary developer, but you were very instrumental in this advanced transcriptomics that allowed for
Yes.
the seeing of the network and some of the phase change of a network. Tell us about what that was in that journey, and just really kind of in your own words, describe what transcriptomics is and the value of it.
Sure. So, DNA is the kind of code book, and RNA is what's read from it mostly. And then the RNA is usually made into proteins or other RNAs that function in the cell. So think of the cell like a factory and the body like a bunch of factories all connected together, functioning. And so by reading, almost every cell has the same DNA essentially, to the first approximation. But what's different about a liver cell and a kidney cell and a brain cell is what genes are turned on, how that DNA is read, which chapters are open, which pages are turned on, which words are actually read off the page. And that's the RNA. And so this kind of control of the transcriptome, which is the genome is all the DNA. The transcriptome would be all the things that are transcribed from the DNA, which is the RNA. And so as we began to be able to measure that transcriptome, it became clear that you shouldn't be looking at it, again, one transcript at a time. What people were doing initially is they'd compare things. Like, let's say you took a neurotypical brain and an autism brain, and people had died, and you had postmortem tissue, and you wanted to compare the tissues. So you could look at the transcriptome, which is all of the RNA expressed, and compare them. And people would look at what's the most up gene, what's the most down gene. And that just didn't seem like a very complete way to do things, right? It didn't make a lot of sense to me. And some of my other colleagues, including Steve Horvath, who developed the statistical framework that we use. And we worked on that quite a bit together early in our careers. And so, essentially, the issue was we thought that by looking at the transcriptome, it's giving us an unbiased view of the state of whatever tissue we're looking at. And it's telling us what's different about it and what's same without any preconceived notion. Now, you could say that, oh, you're not being hypothesis driven, but that's just not true. My main hypothesis is that there are thousands of hypotheses you could have. How do you know which one is right? Biology is not physics or mathematics. It's not a pyramid of knowledge. We're still in this highly descriptive phase. And so when we do biology research, we're not testing theorems and things that are well established. We're describing, quote unquote, mechanisms. So the only theory, Dobzhansky, said this well, I always misquote it a little bit, but, biology only makes sense in light of evolution, right? In the context of evolution. So that's the only theory we really have. So we're really describing things. And so our thinking was, well, if we're going to be doing this, I want to have a way of prioritizing hypotheses in an unbiased way. So I want to be able to look at all of the data. I want to be able to look at all the genes and for it to tell me what are the major biological processes that are altered in a condition or in any experiment that I do or in a disease? And how does that relate to the underlying genetic risk that we know as well? That's critical. Because measuring the transcriptome, which is RNA, is like measuring anything else that geneticists call phenotype. You could be measuring blood pressure or height or weight or your metabolism or your glucose. Those are all called phenotypes, or brain structure even. It's hard if I find something different between autism and schizophrenia or something and neurotypical people who don't have those conditions. Is that caused by having the disorder, or is that causal? That's always the question. Where's the causal arrow go? Genetics gives you a causal arrow. The disease doesn't cause the thing that you inherited. The thing you inherited is the starting point. And it may be interacting with environment and development over time. It can be very complicated, but at least it gives you a causal anchor. So we started to look at the transcriptome, and we started to measure it. And we kind of reasoned that neurologic disorders like stroke, multiple sclerosis, Alzheimer's disease, all the dementias really are different from psychiatric disorders. They're considered differently because they have visible pathology, either under a microscope or even visibly, like with an MRI or in an autopsy. Whereas psychiatric disorders have no visible brain pathology. And we reasoned, well, now we have these tools that are essentially a much stronger microscope where we're looking beyond cellular resolution. We're looking submicroscopically using sequencing of RNA and the ability to measure that. So it's kind of as our microscope, you could think of that as an analogy, has gotten better and better. Our ability to see has gotten better and better. So at this level, looking at RNA, we reasoned maybe looking at the RNA will tell us something about autism or psychiatric disorders in general. And so we did this in autism for the first time. It was published in 2011. And we showed that patients with autism had changes in RNA in their brain compared to control patients that couldn't be explained by confounders related to autopsy or age or anything like that, or seizures, that really differentiated them from controls. And subsequently, over the next 15 years, we've been able to validate that quite extensively in many more cases than the original small sample size. And we've also been able to then take what's known about the genetics and ask, of the changes that we're seeing using these transcriptomic network methods, which of these are being driven by genetics and which are likely to be secondary or signs of resilience or signs of susceptibility or just part of the disease that isn't the initiator. That doesn't mean they wouldn't be good treatment targets. They might be, but they're not initiating. They're not related to the cause. So that was a really surprising finding because, we were looking at that time at 19 or 20 cases of autism. We knew one of them, we knew what its genetic cause was. It's a rare form of autism called 15q duplication. But the others were unknown, but we assumed what we knew about autism, that they all had slightly different genetic causes. Most of the genetic causes of autism that are known are rare. And so if you had 100 cases, you might have 100 different forms. And so it was surprising to see this kind of convergence, this kind of coherence across 19 cases. Not all of them had the pattern. I would say it's seen very strongly in about 2/3, a little weaker in 3/4, and maybe not seen in about 25 to 30%. So that's what we kind of know at this point. And now we've validated it in much larger sample sizes, and we've even used more modern techniques to look at it from different angles. And now we can do single cell transcriptomics instead of bulk tissue. And what that allows us to do is to connect these alterations that are occurring to specific layers of the brain and specific circuits and specific connections that are going on. And again, what that's showing us is that the most susceptible regions, but not the only ones, are the regions of the brain that are involved in connecting to each other. Not that, there are issues at the local circuit level too, but the most profound things have to do with things that connect the hemispheres and that make long-range connection between regions, which fits with the imaging literature, which shows that there is some of this long-range disconnection in some patients using functional imaging. And so, if you look back at the last 20 years, we've gone from identifying genes to identifying the molecular pathology of the disorders to now having a first level understanding of what are the most impacted cells. And now the job is to understand what that actually means functionally and how we can fix that. Another thing we can do is once we have a transcriptome, we can ask, well, do other disorders have similar changes? Where they overlap, where they different? And so we did the first two major studies comparing autism to schizophrenia and bipolar disease using the transcriptome. One of the nice things about that is that there are very few clinical tests that you can do across a 2 or 3-year-old or 4-year-old with autism, a 20-year-old with schizophrenia, right? Or somebody with bipolar. Like, what are you actually measuring there? Here, we're getting down to, by measuring the transcriptome, we're measuring the cell composition and the changes within the cells that are actually occurring. So it's giving us a first look at potential mechanisms of disease, where they might overlap and where they differ. And they mostly don't overlap, right? They're mostly different, but they have areas of overlap. One of the strongest is in changes in excitatory neuron function in schizophrenia and autism. But that's still at its very early stages, those kind of cross-disorder comparisons, but they're very intriguing.
Yeah.
There is some shared liability across schizophrenia, autism, and bipolar, but it's not the majority of the liability. It's some shared liability. So that's important to know as well. But we also know that these disorders are quite distinct when we look at the molecular pathology.
Yeah. It's so interesting. And you mentioned early on, depression or other kind of mental health issues or conditions. I mean, what does that tell us? What might that tell us about a broader sense of, because I think you said about 50%, less than 50% is genetic. For like
let's call it
depression.
50%.
Man.
Yeah, it doesn't mean, it means like, inheritable, and there's a big gene environment interaction there. Yeah.
Right. Yeah. So do you think there will be kind of breakthroughs in that understanding of mental health, where, because
Yes.
we're already kind of moving on from, oh, that's just feelings and, but there's probably something going on that's very physical. Where do you see that evolution in a broader sense for mental health? Depression.
Well, I mean, depression is very complicated, and I'm not an expert in it, but I'll say a few things. One is that, depression is way more common than autism, and one has to differentiate between getting depressed because of a situation, which almost anybody has felt depressed, versus real major depressive disorder, where somebody is depressed for a prolonged period of time and has all the vegetative symptoms, and somebody who might have this recurrently over their life, where it becomes a lifelong disorder. That's very different. So we're beginning to understand with the genetics that those things are really different. And so I think, depression is probably even more heterogeneous than something like autism, which is very heterogeneous. And it's also more common. So it's challenging to study. I do think that as we get larger and larger sample sizes, right now, in my view, there's not an incredibly clear molecular pathology of depression yet, or subtypes. But I think we're at a stage where we could bring all of the data together, and we might be able to get a better understanding. So I've started to talk with colleagues about that. But in some ways, it's a harder nut to crack. On the other hand, it's also something that's remarkably treatable in many patients, not everybody. You could say in half, or, a lot of people respond to the current, but there still are many people who have recurrent depression that's refractory, and that's really terrible, and obviously suicide connected to that as well. So we have a long way to go there, but it's really in a different place.
Yeah. So you said another thing that you said I found really interesting, because I did not know that there were people that, a very small amount of people that are born with a brain that doesn't connect the hemispheres. And I forget what the condition is, but
Agenesis of the corpus callosum.
So interesting. So, but they're functioning, and you really don't know till you really kind of look, or
Test them.
Yeah, you test them. When you say the transcriptomics allow for us to now kind of look inside the cell and then come back out and look at the network of the cells inside the brain in that larger network, what do you think we're going to find as we continue to really understand that larger network, and how the different hemispheres in the different places work, as well as make the whole brain resilient?
Well, I'm going to cop out for a second and say something that's really important to understand, is that we have no model for how the brain works that is really useful. So we don't know how the brain works. And the question is, does gathering these data in many conditions where it may not be working optimally, is that going to help us develop a model for what it's actually doing and how it's working? Hard to know. Like, can you figure out how a car runs by just breaking it down in many different ways? Certainly that will help you understand what the individual parts might be contributing to. But getting a whole philosophy and rubric of how the whole thing is working and what it's supposed to be doing may be a little bit tougher. Of course, a car analogy is trivial, and I don't mean to extend that metaphor very far. The brain is obviously quite different, and so that metaphor only goes about this far. So I think that's a major problem, is that if I find a gene that's mutated in a patient with a dilated cardiomyopathy or an arrhythmia or high cholesterol or some other immune condition, I can connect it very quickly to some functional pathway, to some tissue that fits within a model of how that organ works. And I can have a first or second level understanding of what that mutation is likely doing. In the brain, I can find things that affect a channel, it might affect neuronal firing, and understanding things like epilepsy might be a little easier. A little bit. It's still very complicated. But understanding how it contributes to social cognition or intellectual function kind of is a whole nother issue. And again, we have cellular models. People have gotten Nobel Prizes for some of these things, but they're still pretty far from actually having a model like we do for how other organs work. And that's one of the really big challenges, I think, that we have to face. Maybe the genetic revolution and all the omics that we're doing, these transcriptomics and things, will kind of circumvent that, so that we'll just be able to repair that. And that may be sufficient without even fully understanding how the whole system is working. Kind of like the way dopamine was identified without understanding how the whole basal ganglia worked and supported movement. Of course, Parkinson's disease and other disorders of the basal ganglia is what drove us to really understand how these things work and how these circuits initiate movement and help movement. And so maybe in some ways, these areas of social cognition and repetitive behavior, we'll begin to understand how they come about more by understanding how they're broken.
So how did you get on this path, professionally, where the study of autism and bipolar and in schizophrenia, and why, what was it, kind of that pivotal moment there for you as a professional, that's where you decided this is where I want to kind of throw all my research and my effort into?
Yeah, what happened early on in my career, I was working on brain development. I've always been interested in the brain development and language and brain evolution. And so the question, I was fortunately introduced to folks who had just started a foundation called Cure Autism now, Portia Iverson and John Shestak. And I didn't know much about autism at that time. And they, we hit it off. I realized that what they were doing was fantastic. And even though I was just starting my laboratory, I saw the huge unmet need, and I saw it as a place where I could have impact potentially. I think in this kind of career, you go into it to do something significant, not for you, but for the world in a way, to have a significant life in some ways that impacts patients. I'm a physician, and I take care of people on a, when I see them, but there's a limit to what I can do, especially as a neurologist. So I focus most of my career on research so that we can change the way we treat brain disorders. And so I saw it as it was an unmet need. There wasn't a ton of research going on that at that time. And so I got very involved with them and setting up a gene bank for autism, because they asked me, what should we do? And I said, well, this is like a really heritable condition. We should motivate genetic studies. And in fact, we shouldn't just motivate people who are in the field. We should make this data bank available to everybody so that we increase the field. And so we created this thing called the Autism Genetic Resource Exchange, which, was the major resource for autism genetics for around a decade. And that led to many, many findings and hundreds of labs getting into autism genetics. And that's kind of how I got into it. And then I saw using, that people weren't integrating data enough. There were people doing standard kind of genetic approaches, but there needed to be a bridge between identifying markers in the genome that are related to disease, identifying the actual genes in the genome that are doing that, and how those genes work together in a neurobiological system in the brain or in the tissue. And so my work has been kind of connecting all of that. Necessary
And fulfilling, I'm sure, because you're pushing the limits out there. It's not just the technology that we get to better understand. It's these questions that you're asking, the connections between these areas that are really kind of pushing back the science.
Yeah, one thing that kind of surprised me early on, there's incredibly brilliant statistical genetics people who were running these genome-wide association studies, identifying what they thought were genes, but they weren't. They were just identifying markers in the genome. And they didn't really understand, I felt at that time, based on their questions and their attitudes towards the work that we were promoting, that you needed to do a lot more to understand the function of the genome, to understand what was actually the causal genetic variant and in what genes it was regulating, and then what that means in terms of developmental networks as the brain is developing, and what that means in terms of cell types, circuits, etc. And all of that needs to be connected to have some understanding that that step of GWAS or finding a gene is just really a first step. It's a first necessary step, and it's been transformative utterly for the field without a question, because it moves from just measuring things to identifying causes, which, again, gives you a critical starting point, but it's not enough in and of itself. And I think people are realizing that now.
Yeah. Yeah, and that's all been in the last, 15, 20 years with the, mapping
Yes.
the genome. And
Yep.
it's really incredible when you kind of step back how far we've come and how fast we've come.
It is amazing. If you had asked me 25 years ago where we would have been 15, 20 years from now. Even 10 years ago, I wouldn't have been able to predict how fast we would have moved the field.
Yeah.
So it's really a testament to technology, but also a lot of more people coming into the field, a lot of more people working on it, a lot of people realizing how important it is. All of that worked, but a lot of it is really technology, ability, know-how. And, one of the things I was told early on was you need to work on something simple that's solvable in 25 or 30 years, and by some very smart people. And that's true, but I was told not to work in things like autism because it's too complicated. How are you going to, looking at RNA in the brain, all the RNA, how are you going to get anywhere? So first you have to have organizing principles, right? And that's where the gene networks come in and, and some of the other integrative genomic techniques, so you can begin to understand cause and effect and the relationship of one gene to all the others. But my feeling at the time was, well, if I don't do it, who's going to do it? If people don't start chipping away at this. And I guess I was lucky enough that the technology was there. If I had started it 50 years ago, it might not have gone anywhere.
Yeah.
So I think there's a kind of coincidence of technology, the field, know-how, funding, everything kind of came together to make this kind of work. And now the field is really quite large. We kind of introduced this term convergence of in autism or other psychiatric disorders. And one of the reasons for that was, in modern biomedicine over the last 200 years, disorders are defined using some kind of what we call pathophysiologic framework. That's the pathology, what you can see in the tissue, and the physiology, which is mechanism, kind of connecting those together. So the whole idea is that you go from kind of cause, the thing that initiates, over time to see how a disease progresses and percolates and actually becomes a problem over time. That's the pathophysiology, the kind of natural history of disease. And so, the kind of notion that psychiatric disorders don't have a pathophysiology never made sense to me. They're brain disorders. And so, the idea was, well, to define pathophysiology, let's see if we can identify some molecular pathology, which is another kind of foundation, of everything. So, in modern biomedicine, cancer is a disorder of proliferation and invasion. All cancers kind of share that to one degree or another. Some are way more invasive than others, some proliferate more, but they share that. Now, that's a kind of broad pathophysiologic framing. Of course, when you want to understand an individual cancer, being able to dissect it at a molecular and genetic level gives you far more information than just understanding what I just said about pathophysiology. But the pathophysiology is like a starting point where now one can understand how do these mutations lead to the proliferation and invasion. There's a framework within this kind of pathophysiology in which the biology and genetics is interpreted. And that's where we're trying to move in these psychiatric disorders. And again, the problem is we don't really know how the brain works. So but we know pretty much how every other tissue works, or at least we have working models that are that seem to function. So it's an interesting conundrum that we have. And I think there will be breakthroughs here where finally some treatments will work, and that will tell us that we're on the right track.
Yeah. So what do you tell the young researchers in your lab or those that you mentor? What advice do you give them, especially as you think about maybe the next 10, 15 years and the possible convergences or the speed up, if you would, the acceleration of some of the learning? And what are you telling them where to kind of target, look for, kind of gear towards?
Sure.
What are you telling them?
Well, it's a really, really great question. I mean, it is. So one of the things that I've been pushing for, 20 years is the importance of having kind of some quantitative data science approach in neuroscience in general. In that biology is becoming more and more quantitative, and the better you are at statistics and analysis, the easier it's going to be. Another thing I tell people is it's really critical to use as much of the data that's out there to frame your experimentation. Before you spend a year doing an experiment, why not see if somebody else has done it, or that you could do it on the fly in a couple of weeks using data? And so that's really important. My main focus has been in developing high throughput biology. As we gain more and more genetic knowledge, we have to study more and more genes, and we have to study them in parallel to really get a good understanding. So building systems where we can study human genetic variation at scale and understand how it impacts human neural development, maybe using in vitro model systems like stem cells, which we've been doing, is kind of one way to go about this. So we've been really pushing this, but the future is going to be a lot of data analysis. But there's nothing that supplants old-fashioned experimentation for proving something. And so I think there's an iterative process where one does analysis, comes up with hypotheses, orders them based on the data, tests them, revises the models, etc. And that's going to get faster and faster and faster. And that's really exciting. And it's possible that a lot of experimentation in silico, like we'll have enough data so that the models can be done in silico. That's possible.
Yeah.
Yeah, I don't know if that makes sense.
Yeah, it does. It really does, because what you're saying, I think, is important too, is that there's a whole bunch of different relationships that researchers are going to have to have. I mean, it's not going to be enough to just have partnerships or relationships with other researchers in biology. You're going to need to have,
Oh, absolutely.
those that understand, quantum or computing or machine learning or.
I mean, that's already happened.
Yeah.
And so we have really fabulous collaborations. I have a autism data science grant with Mark Gershwind at Yale, who's a computer scientist and bioinformatician in his lab. And we've worked together for many years on PsycEncode, building, initial models of the brain. And again, those types of collaborations are essential. And I have other collaborators here at UCLA. It's one of the things that makes a place like UCLA very interesting, in that it's a major medical center, but, we're right on the campus. So I park where the engineers park. And we all talk with each other. And so we have the ability to do the kind of multidisciplinary work that's really necessary.
Yeah. Yeah, that's great. So our time is, unfortunately, it's come to kind of pass here. Just one last kind of thought. What is your greatest hope for the future here? When you look out, what do you hope to, in the next 5 or 10 years to really kind of figure out and kind of leave that on the table?
Yeah, a couple things. So one is, we also, we work in autism, but as we developed all these tools in psychiatric disease, it made me realize that neurologic diseases were syndromes just like psychiatric diseases, where it's just that because they have pathology, people assumed it was one thing. And that's not the case. And so we've also applied these methods to neurodegeneration, especially conditions like things that involve this protein tau, like Alzheimer's, frontotemporal dementia, and progressive supranuclear palsy. And we're fairly close to having drugs, that we've developed based on some of these methods, that we're working through to see if we can get them to move towards clinical trials. So I'm very excited about moving into that. And the same thing with autism. I think that we have some initial proofs of principle that we can turn some rare genes back on. The question is, how do we engineer that and move that into people? That's going to take large groups working together. That's something my laboratory can't do. I'm not even sure it's something that a single biopharmaceutical company can do right now. I think it's something that requires team science. And so those are the kind of things that I'm really excited about. The next decade is actually moving some of these genetic findings into therapies, or at least moving them forward towards the therapies, both in the neurodevelopmental and the neurodegenerative side. And we're a little closer actually with the neurodegenerative. In some ways, it's a little bit easier because when we're screening drugs, we know what we're looking for. We're looking for things that stop neurons from dying, because that's what happens. Right. So, I mean, that oversimplifies it. It's way more complicated than that. But so we've been going back and forth between these different areas, repairing the damaged nervous system, neurorepair in health and in dementias, as well as the autism, because they complement each other. We can test things in one that we can't test in the other, and then they inform each other. So I'm kind of excited about where we are there, because I'm really seeing that synergy, and I can see in the next 10 years we will be moving towards drugs. The last thing is to see if we can use these gene networks without understanding the mechanism to find drugs. We can use AI to connect gene networks to drugs and then see if the drugs reverse the patterns back to normal. That's actually ongoing now. There are several small biotechnology companies doing this and others, and it's something I've been championing for about 20 years, but now seeing that happen is also very exciting. Yeah. So we may be able to move quite quickly. Let's catch up in 5 or 10 years and see if I'm right.
Let's do that. I sure appreciate your time today. I appreciate what you and your lab does and all the people at UCLA do. I think this is amazing, and there's a lot of people out there listening to this that are probably you've given a little bit of hope to, and I appreciate that.
Well, thank you very much. Thanks for the opportunity to talk.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.